guys, so let's look at genetics. Okay, we're going to talk about a lot of things in this intro video. Okay, so let's just jump right in. Some questions that we want to be able to answer by the end of this video. All right, we want to know who Gregor Mendel was, all right, and how he contributed to genetics. We want to know the difference in dominant and recessive genes. We want to be able to describe the term allele and how it relates to different traits in specific genes. We want to look at the words genotype and phenotype, which are vocab words. We want to make sure we know how to use them. And we want to be able to do a simple Punnett square. Let's look at Gregor Mendel. He was an Austrian monk that studied inheritance in pea plants. So he looked at how traits were passed on from one generation of pea plants to the next generation. He did have a background in math, and he used statistics in order to describe how traits were being conserved or being passed on. He is the father of genetics. We want to make sure that we know his name. So Gregor Mendel is the father of genetics. Now, when we talk about the different traits that he looks at, or that he looked at things that were observable, right? So he looked at like the length of the stem or what color the flower was. He looked to see whether, the pea pod or the pea itself was wrinkled or it was round, or if the pea pod had this like pinched edge or if it had a rounded edge, right? So he looked at things that he could observe. And he started blending these traits, right? He would make sure that, one parent had, say, a wrinkled pea while the other parent had a smooth pea or a purple flower compared to a white flower. He was surprised to find out that those traits didn't blend, okay? The offspring either had one trait or it showed the other. And that led him to call traits that always showed up, no matter the parent, or no matter which, if both parents had it or if only one parent had the trait, dominant traits. So they are more powerful. They are going to cover over the recessive traits. And obviously the recessive traits are traits that are weaker. So when we write genes, okay, this is something that you're going to see in this video. So let me just talk about it real quick. You want to look for capital letters versus lowercase letter, because that's going to tell you if something is dominant or if something is recessive. Now, what is that going to look like? Well, let's say we wanted to write tall as a set of genes, okay? So we would write big T for tall, and the opposite of tall would be short. You might think, well, we'll write S. Not true. If short is the recessive gene, we would write a little t. So a parent, right, because they're going to have two copies of each chromosome, they could be big T, which means they're dominant for tall in a homozygous descriptor. We could be a hybrid, or we could be homozygous recessive for short. So in dominant traits, you would see tall here, you would see tall here, and you would only see short if you got both short genes. Okay, so we'll look at that a little bit closer in the next couple slides. Now, heredity is controlled by genes. You get one gene from each parent, right? Because the gametes only have half the parent's genetic code. This is what we learned about when we talked about meiosis, right? So meiosis makes haploid gametes, right? So this term coming back around, haploid gametes. So that's only half of the genetic code compared to their parents. All right. And so the law of dominance tells us if you have two forms of the gene, the dominant one is going to show up. So that's a pretty easy law to remember. A minute ago, we talked about if you had this genotype, big T, little t, I said you would show up as tall. That's because the law of dominance tells us that as long as one of the genes is the dominant gene, that's the gene that's going to show up. So that's why it's really important to look for those capital letters when we're looking at the genes. Now, when you have two different versions of the genes, one from each parent, you're only allowed to pass down one. So let's say we have, here's our parent. It's big T, little t. Well, through the process of meiosis, all right, it's going to make two gametes. It's going to make one gamete that has big T. It's going to make one gamete that has little t, right? Because these are haploid, and this over here is diploid. So the law of segregation tells us that the parent can only pass down one. If this gamete is the one that does or gets fertilized, right? We've drawn an egg here. If this gamete gets fertilized, then that means that this gamete doesn't. So the only gene that gets passed on is that little t. So the law of segregation tells us that organisms only pass down one of its genes. And the last law that we're going to talk about is called the law of independent assortment. And that means that if you have genes for different traits, they don't impact each other. It's not like because you're tall, you have to have green eyes. Okay, they're not related. So how does that, what does that look like? Well, when we're doing meiosis and we'll say big X, and we'll have, we'll say big T, little t. Well, when these guys line up in the middle of the cell, right? So here's our cell. And then through the process of meiosis, right, we end up in metaphase. So we have big X, and we have big T, little t. And these get pulled to opposite sides of the cell. And eventually, right, we know these are going to end up as number 1 and number 2, right, 2 different gametes. Well, just because I got a big X did not tell me which T I got, right? In this one, I have big X, big T. And in gamete number 2, I have big X, little t. They are independently assorted. They're not tied together, right? So that's what the law of independent assortment tells us. Now, we're talking about traits and all this stuff. And I keep saying like big T, little t, and stuff like that. So there is actually a term that we use to describe that, and that's called an allele. So an allele is the version of the gene. So I'm actually kind of starting down here. The allele for plant height for tall would be the big T. Another allele would be the little t. So there's like allele number 1. There's allele number 2. Okay. Those alleles describe traits, and the trait is just the characteristic of the organism, right? So height or color or the P coat, right? Wrinkled or smooth, whatever it is. You can come up with a letter abbreviation for any trait, right? Like, let's say we're talking about mice. We're saying that they have black fur or white fur is recessive. So if black fur is dominant, we give it the big B. And so white fur, we're going to say is recessive. Remember, we have to keep the same letter, so that would be little b. Okay, so that's just a trait. Depending on which genes you get, that's the traits that are going to show up. You can't have something expressed that you don't have a gene for. Now, just to kind of sum up here, okay? Each parent is going to give the offspring one of each of their chromosomes. That's going to give the organism 2 copies of each gene, one from each parent. The gene can be a dominant or a recessive allele. It just depends on what they got from mom and dad. And the organism can have 2 dominants, 2 recessives, or one of each. This is called the genotype, okay? The genotype is the genetic makeup of an organism. So it's a list of what alleles are present. You can either be homozygous, which homo means the same, right? That's what that prefix means. And zygous is talking about the zygote, which we talked about earlier in the year. So zygote is the fertilized egg. It's that first cell. That's where like your stem cells come into, like all that unit that we had earlier. So if you are homozygous, that means the genes that you have are the same allele. So homozygous dominant, big T, or homozygous recessive, which would be little t, little t. Another term for homozygous is purebred or true breeding, okay? Heterozygous, hetero means different. All right. And that means you have one of each. So big T, little t. And a lot of times we just call that a hybrid. All right. Hybrid means a mixture of 2 things. And so we call heterozygous hybrid. The phenotype is what you actually see when you look at the organism, like when you make an observation with your eyes. So that's the version of a gene that gets expressed. And the phenotype is going to describe what allele of the trait's being expressed. So if you were to look and say, okay, what's the phenotype for these flowers that I have on this slide? Well, the phenotype is pretty simple. It's either purple or it's white. 3 of the flowers are purple, 1 of the flowers are white. That's the phenotype. And a lot of times we'll talk about something called the phenotypic ratio, which in this case is 3 to 1, 3 purple to 1 white. Now the genotype is different. In this case, the genotype is 1 to 2 to 1 because we have homozygous dominant, which is the 1. We have hybrids, which are the 2. And we have homozygous recessive, which is the last one. So genotype can be very different from phenotype. Remember, even though that there is a recessive allele there in those we'll say 1 and 2 flower, the law of dominance tells us that the dominant gene is going to show up. And when you're determining genotype, that can be difficult, right? So if we go back and we say, okay, we're going to pretend that we don't know these. So we're going to cover those up. All right. We say, okay, we know that purple is dominant. So there's purple. We know that little p is recessive for white flowers. And I say, okay, guys, here's the phenotype. Tell me the genotypes. Well, you can do one of them, right? The homozygous recessive one is easy. We know that one's little p, little p. But these 3, that's where we can get a little bit tricky because it could be big P, right? Could be big P, or it could be big P, little p, right? So determining the genotype can sometimes be difficult. If you have a hybrid, then you're like, well, the little p is covered up, right? I don't know. So in order to figure this out, we do what's called a test cross. And this is something that we would do in the lab. We would take a plant and we would cross it with another plant that we knew the genotype of, and we would see what their offspring looks like. So, do we end up with all tall organisms, right? Do we end up with all short organisms, something like that? And we could figure out what the genotype of the parents are. Now, there's a probability that is associated with this, a little bit of math, right? So some probability you're probably already familiar with is coin flip probability, right? You flip a coin, might get heads, might get tails, but it's pretty much a 50-50 split. Since the parents are only passing down one of 2 genes in our simple discussion of genetics here, then we can also look at some similar probabilities. So when we do a parent cross, the outcome of the parent cross is going to be described using percentages. So let's look at one of those, all right? We're going to use T, big T for tall, little t for short, and we're going to cross a homozygous dominant, that one, that's homozygous dominant for tall genes with homozygous recessive. We're going to put in our Punnett square and we're going to predict the genotype and the phenotypes for the kids, for the offspring. Now, each possible gene for the parent goes above one of the squares on the top. So you guys have, I hope, seen a Punnett square before. It's going to look something like this. So each possible gene goes above one square on the top. So we have our homozygous dominant above this square. The second allele goes on the right square. And the same thing on the sides. We have little t on the side. You can only put 2 letters in a box, and you just kind of fill them in. So this T here on the left goes down, down, and the little t goes in, little t goes in, little t, big T, big T. All right? So there we go. Each box is worth 25% of the offspring because you have 4 boxes, right? So 1 out of 4 is 25%. So this is the Punnett square that we get, all right? And in this case, if we wanted to look at the percentages, we would need to write those out. So we have big T, big T, big T, little t, big T, little t, big T. Oh, sorry. Where's my eraser? And little t, little t. This is called a dihybrid cross. All right, how do I know that? Well, I'm looking at the cross, right? This X thing right here tells me I'm crossing parent 1 with parent 2. All right. And what I end up with is 25% big T, 50% big T, little t, and 25% little t, little t. Now, there are a lot of different ways we can express that. We can write it like this. We could say this is a 1 to 2 to 1 ratio. And it's implied that we're looking at homozygous dominant, hybrid, and then homozygous recessive. Okay, that's the order. We always write that in. We could also talk about the phenotype, right? What's the phenotype here? Well, I've got 75% tall. I've got 25% short. It's important when you talk about phenotype to use descriptors, things that you would actually see, right? So like tall and short. When you talk about genotype, then we actually list the genotype with the two alleles, right? So this would be the genotype. Now let's compare that to the first one we did. Here we've got 100% hybrid. So big T, little t. That's our genotype. We have 100% tall. That would be our phenotype. This is a homozygous dominant crossed with a homozygous recessive, right? In our dihybrid cross, we get our 1 to 2 to 1 ratio. Now, there are a lot of different things we could cross, right? We could do, we'll say a little t parent with a big T, little t parent, right? So we have a hybrid and a homozygous recessive. So I've got big T, little t, big T, little t, little t, little t, little t, little t. All right, so take a minute, pause the video, and come up with two things. I want to know the genotype, and I want to know the phenotype as they are described by ratio. So hit pause, try to write them out. When you come back, we'll do the answer. So I'll give you just a second. I'll drink some coffee. Okay, so the genotype is 50% hybrid, 50% homozygous recessive. The phenotype is 50% tall, 50% short. All right, so that's always what we want to look at. We could do the ratio, 1 to 1. Same ratio for phenotype, 1 to 1, 50-50. Okay. Now, the more generations that you cross, you have to have a way to kind of identify them. So the P generation is called the parent generation. This is my parent generation right here. In this case, I had big T, little t by little t, little t. There would be my parent generation. The F1 generation is the first generation of offspring. So all of these guys right here, these would be F1. Now, if I did another Punnett square and I used the F1 kids as the parents, we would call that outcome F2. So we had, we'll say we used this one and this one, right? So now I'm crossing a homozygous recessive set of traits. I'm going to get little t's everywhere. So this was F1. So now all these guys would be F2. And in this case, I would have 100% little t, which would be 100% short. That would be my genotype, and this would be my phenotype. All right. F3 would obviously be the next generation. Okay. So hopefully you can go through and answer these questions. We're going to look at some more examples of Punnett squares and genetics in the next couple of videos. This is a great way to start. So make sure that you can answer all of these questions, and let me know if you have any issues or concerns. Send me an email and I'll be glad to give you a hand.